To be frank, I'm not sure that it is worth to broadcast this through Zoom because I'm not sure that anyone is looking at us. But anyway, so it's not a problem. Okay, great. So I would like just to go through this thing. Can I make a note? The first day there were some paper pads, but they disappeared. So I would like to, yeah. You give me only half. Okay, so you said that Pranati talk, right?
Yeah, she is already presented.
Okay. Pranati, all right. Okay, any other that, so Abdul left, Tejas talk, Pranati. Okay, then team number 2 is doing very well as well, right? Okay, good. Then team 3. Mutu, who is Mutu? He left. Mutu also? Okay, then we are. Okay, so I will check it out in my files because I'm not sure that I have everyone's presentation. So that means that the only one that failed was Abdul, right? At the end of the whole thing. Okay, so I'm sorry for him, but anyway. All right, so I would like just to discuss a little bit about this IGM thing, just to send you, to give you specific instructions on how we can structure that, okay? So, well, first of all, as this will be, the presentations will be the last day, what I intend is to be a kind of a happy moment, you know? So it's not for suffering, it's not for being nervous, it's just for kind of celebration and for sharing your possible creativity about ideas that may emerge out of having listened to all the stories that I have been telling you these days. So the idea, I so far I have like 4 or 5 expressions of interest. I will wait until tomorrow, but you know, I think that the ones that have responded me until that point will be the ones that will be doing something. The others, I think that will be impossible to fit because I have to, well, maybe we can fit them, but you know, the thing is that if we have like 4 or 5 presentations, then each of you will have some time for discussion and so on. If you have 20 presentations, then things can be a bit more complicated. So one other possibility that you may entertain is that instead of having a project, an individual project, that you decide to have a project together with some of your friends or colleagues or something. Okay, that's perfectly fine. And sometimes, you know, these projects are more, say, interesting if you have them as the result of a discussion. But you prefer to do it individually, is also perfectly fine. So the idea is to listen to what ideas you may have, these talks of these previous days have inspired in your mind, and whether you can propose on that basis something interesting. So as I mentioned in the message I sent this morning, the idea is that you prepare a few slides. They don't have to be super complicated, but just a few slides explaining what is the general idea, and this is the kind of more academic part of the thing, what components, what parts you think that you need to put together what you propose. Then you explain what would you expect, or how you expect it to work. And then finally, it would be nice if you can make some kind of side comments on what could be the societal consequences or the benefits or the downsides of the technologies and so on that you may share with us and with your colleagues in this class, okay? So I think that that's something that if you have any doubt or something, just ask me. And as we agreed yesterday, we will spend for the rest of the 3 days that we have left for that, the 3rd hour of our meeting just for you to spend the time trying to polish your presentations and so on, okay? So let us do that. I think it's important that if you have not done it before, please have the curiosity to go to the IM place, find the section of projects, take a look to them, see how they look like, and then on that basis you can get inspiration on how you have to present your aspect. Okay, so I think that that's basically what I wanted to tell you today. And so in terms of pending presentations, we have today your presentation, and then on Tuesday we have Sunko to talk to us on Tuesday, okay? But for now, let's have our friend talking, okay? Sure.
My presentation will be a review of this paper. This is basically about standardization of biological devices. So this paper takes a kind of hardcore engineering approach to biological devices. We will jump right into the paper. I will first go into what this paper basically proposes. So this paper proposes to adapt existing frameworks for describing engineered. This paper proposes to adapt existing frameworks for describing engineered biological devices to biological objects, and the objectives of the same are to direct the refinement and use of biological parts and devices, support research on enabling reliable composition of standard biological parts, and to facilitate the development of abstraction hierarchies that simplify biological engineering. I think those of you who are from engineering background should be aware of how we use parts. Like if we are going to use a parts, we have a data sheet, and we just refer to the data sheet to get all the information about the parts and the compatibilities and stuff. So this basically proposes to do the same for biological devices. We will see the same in the next few slides. So this is the device they have constructed as a proof of concept to show the entire idea of you having a standard data sheet and using standardization in biological devices. The previous framework that I showed, it kind of uses that to describe one engineered biological device as a genetically encoded cell-to-cell communication receiver. The description of the receiver is summarized by a datasheet, like I just said, similar to those which we use in engineering. The process of refinement and characterization leading to the datasheet will serve as the starting template for producing many standardized genetically encoded devices. So basically, if you just want to produce another device and incorporate this device into that, you just have to refer to the datasheet and you will get all the information from there. And going just a little bit into the details of the device, you can see it on the right side of the slide. The mechanism and function. It is a transcription. It uses a transcription factor LUXA that is active in the presence of the cell-cell signaling molecule, and this molecule is basically the signaling molecule we are using here, and it is controlled by a regulated operator. So the input is that molecule, and the device output is in form of POPs from the LUXA regulated operator. So it uses POPs to basically standardize the entire thing further, because that can be kind of taken as a common point for any biological device you are using. And in this paper, it has been used to produce a Boolean AND function, but the paper goes into further details and says that we can also use it to create an amplifier. And I will be going a little bit in details over some of the things that they have aimed to standardize to and give details in the datasheet to provide better interoperability between whatever you are planning to use this in and the device they have described. So a crucial measured characteristics is the transfer function, which is the static relationship between the device input and the output that allows the prediction of equilibrium behavior of composed devices. And the dynamic behavior of the device is often reported so that the response time of the device can be compared to the expected timing of the overall system. Now, the response time, I feel, is a very important factor here, because this is not a thing we see very well defined in most biological papers. They have the data, but they kind of gloss over that data very quickly. So here they have given the response time, and from engineering perspective, this is very important to actually use the device in something else. Also, it is important to report the compatibility of the device function with other devices or different operating conditions, wherever the context in which the device operates is expected to vary. This is very much like any electronic device or any electronic component you will see. They basically have the entire compatibility details of the device given in the datasheet. Also, the reliability or expected failure of the device. Also, the reliability or the expected failure time of the device is given, and it is relevant wherever correct device performance over longer time scales is required. This is also something we don't very often see in biological papers. The experiments conducted are generally over a very short time scale, and this question I think has been raised several times here, that what happens if we continue the experiment? Does the same behavior persist, or does it change? Does it mutate, or the medium no longer supports the organism? And finally, a description of the power and material resources consumed by the device informs the choice of suitable power supply and resource pools for the system. And this is also very much like engineering devices we used, rather than taking approach that is more common to biological papers. They have also outlined some characteristics that will enable easy use of the receiver in other applications, and some of them are use of standards that suppose the reliable physical composition of genetic parts, a device design that produces an output signal that is a common signal carrier. The common signal carrier here referred to being is POPs, and you can actually backtrack and calculate POPs from the cell signaling module or whatever product the device is producing, and that is basically the only approach I have seen that is being taken in the papers. You backtrack and calculate the POPs value, and if you have a datasheet like this, that might be a possibility and pretty easy to implement. And they have also given an extensive qualitative device description. I am not going into the construction of the device in details because I can't explain that to you. You better refer to Professor Lorenzo for that. And then we will get into why standardization is necessary. So, synthetic biology is a field where both biologists and engineers are working. And engineers basically expect the data sheet. When engineers are working on something, they are using a device, they expect to refer to a data sheet to see the specs of the device and the compatibility. And also from a experimental perspective, this gives you an overview of what the device is going to work with. So you don't have to do back-to-back experiments to figure that out, and you have a basic thing to refer to. You don't need to spend more time in experimentation. That speeds up the entire process, and like Professor Lorenzo said, the technologies are getting outdated very fast. So for actually producing some useful research, this will be very necessary. Actually, this is very necessary even right now. Also, standardization makes the entire process of getting approval for your genetically modified organisms easier. It speeds up the approval process, and till now we have seen that in respect to genetic chassis. And if you are also standardizing the components that you are using the chassis, this will probably make the entire process even faster. Now I will go into the merits and demerits of the paper. I could not see any apparent demerits of the paper because they have taken an engineering perspective and they have gone very deep into that. Their data sheet almost covers all the data that you might need from any device. And as per the merits of the paper, this basically provides a framework for what the spec sheet for a biological device should be. And it kind of establishes standards for how you should define the devices that you are using or the devices that you are creating so that they can be used in other applications. Thank you.
Well, you already mentioned merits and demerits. So, in my view, the authors of these papers are what you may call hardcore bioengineers, in the sense that they are engineers. They don't consider things that they don't want to consider, and they ignore phenomena such as evolutionary problems, mutations, context dependencies, and all that, right? So they just take for granted that you can take a part, standardize it, it will work, you have parameters, and that's it. And one of the features of this art, on the one hand, is really interesting that they propose this type of vision and very disruptive. But at the same time, I don't know whether it's completely fair that the authors ignore all the knowledge on the context dependency of biological systems or biological parts and so on. And this is still an open question, you know. So there's this company in the US called Ginkgo Bioworks. I don't know whether you have heard of it. If not, the last day on Wednesday morning, I will mention that they claim that DNA is the software, cells are the hardware, and that's it. So that means that by changing the software, immediately you generate a new phenotype or a new property or a new material or a new molecule. And, you know, they just they put under the carpet things such as mutagenesis, context dependency, and these things. So I don't know whether you come from a biological or from an engineering background, but, you know, what would be your kind of perspective on that? So
I think that is kind of oversimplification, because for the engineering applications, the devices are much simpler, and we basically understand the entire working of the device. Kind of like if you have an entire metabolic map of a biological cell and you refer to that, we have that level of understanding for electronic devices, but that's not true for biological devices. So ignoring all other things is kind of oversimplification, and I don't know how, I don't exactly remember how they created the data sheet, but it has been created through experimentation. I think it is somewhat valid, but it will, from the perspective of a biologist, it will only work it will strictly work in only those scenarios that they have outlined. If you are using it for anything even slightly different, you will have to run your own experiments, because the thing you said is basically a gross oversimplification of the entire cell.
Okay, and what do you think could be done to move ahead despite the oversimplification? I mean, do you think that oversimplifying things is the best way to go? Or what do you think could be the follow-up of this type of approaches?
The follow-up for this type of approaches should be basically to refer to the metabolic map of the organism to see how your modifications are affecting it. I don't think a complete standardization will be possible in the way that you plug in a device and you get output, because something or the else is very likely to interfere with it. So unless and until you have complete understanding of what is happening inside the cell, it will be very difficult. So another approach we can take is what we are doing right now. We can just figure out what is happening through experimentation. That seems like the only way forward right now for the kind of capabilities that we possess.
Okay, well, thank you. Very good. Any question or any comment to that? No? Okay, great. Well, thank you very much. You did a very good job. I'm curious to know what the chat has. You know, let me see. Let's see. No voice, no audio, is not... Okay, so nothing connected to the science. Just complaints about the thing. Okay, well, there's something interesting in connection to what you have heard today, and is this idea that because of synthetic biology, we can do something that can be very disruptive, and it is production of things at the point of need, okay? In particular from the pharmaceutical industry. You know that, pharmaceutical industry works on the basis of you having a pill, you know? I mean, oh, that's me. Okay, great. So you are sick, you get an antibiotic, or you get a drug, or you get ibuprofen, or you have a more serious disease, you get a still more complicated drug. And well, the way the pharma industry has worked so far is that either chemical synthesis or some bacteria or some microorganism produces an interesting molecule, then it's taken to a fermenter, the fermenter produces it, and at the end you have a pill or an injection or a suppository or something, and you have something that you can buy. This is what the pharmaceutical companies are interested to have, things that you can buy. However, if what we have for today would become a reality, and you have a software and you have a hardware, then it will be, in theory, possible to have a biological platform. It can be coli, or it can be yeast, or it can be some of the simplified chassis. And then the pathways for production of these different medicaments could be just entered into this bacteria, And then you could have, by the same token, that you have a personal computer, you could have in your place and in your house a platform that when you eat a medicament, that you just enter the DNA into each bacteria, wait for a little while, and then at the end you have the product released by the bacteria, and therefore you can take it without having to buy it in the pharmacy. So this idea that appears to be a bit extravagant has been the subject of a lot of, studies, initiatives, and even a great paper by someone called Liu, in which he shows that it is possible to have a kind of a small device with a small fermenter, a small thing for purification of proteins or purification of compounds and so on. And in principle, he even has a picture, if I remember well, in this paper where you have a kind of ambulance, and then in the ambulance next to the place where the patient is lying, you have a device where, not a bacterial device, but an entire device, an entire platform, in which you have a microorganism in which you can change production of different compounds at different times. So that would be a kind of vision. Whether or not it will be possible in the future, I don't know. But then there's another angle to that, and it's this idea of using synthetic biology and this type of programmable synthetic pathways for production for interstellar travel. You know, you go to the moon or you go to Mars or wherever. I don't think I will make it, but maybe some of you will get there. Then you need to have many medicaments, many molecules, and so that you may not bring with you in the rocket, right? So, or you may not depend on the rocket. I don't know if you have seen this movie, The Martian, where someone goes to Mars and then he gets abandoned there and he doesn't. he has a hard time. So if you need medicaments with different type of molecules and so, ideally, you could have that. You had a platform that you could program biologically and you could produce these medicaments in the site of need. I think that would be an interesting vision. To be frank, I don't think that this approach will develop much further because it goes completely against the interest of pharmaceutical companies, because pharmaceutical companies just want you to buy the thing, right? Another follow-up story in connection to that is this idea of therapeutic probiotics, right? So there's this notion that in the future, because of synthetic biology and these possibilities of programming things and everything, then we can have our intestine, our gut colonized by a collection of engineered bacteria that will sense that something is wrong in your body, and as a response to that, will respond producing a compound that will counteract the reasons why you are feeling bad, okay? So, we're not there yet, but still it's an interesting vision. For the time being, what we have, and again, I'm sorry that we will not have much time to discuss synthetic biology applied to microbiome engineering. This is an incredibly exciting area of development for synthetic biology. But for the time being, what we have is the possibility of having therapeutic bacteria for some specific diseases. And there are various issues connected to that, but now the possibility exists to use engineered bacteria as probiotics that go into our body, and then they eliminate some pathogens, or, well, you have a number of possibilities. So these are new avenues that obviously, to bring that about, require to really fix and to deal with the problem of stopping evolution, stopping generation mutations, generating more genetic stability and so, and these are still open issues. And one thing that is a bit frustrating in the synthetic biology literature is that all these problems literally are put under the carpet. No one wants to talk about them, right? And eventually they may be super important to eliminate, to really make the promise of synthetic biology a reality. The last day, when we had this conversation about what has been your reaction to this course, I will be very interested to have your opinions on what you think should be investigated, on what you think are the weakest parts of the entire story that you have heard these days, to make it real, okay? So is it context sensitivity? Is it controlling mutations? Is it evolution? Is it, I don't know, you name it. But please think on what you think. Imagine that I have a lot of money, and you have to tell me in what should I invest my money to make synthetic biology and to deliver the promise of synthetic biology in reality, right? So or maybe having more mathematical models, or having more tools, or having more parts, or I don't know. I mean, there are many things. And if I were the president of a granting agency, what topics you think should be fixed before the whole narrative of synthetic biology becomes more convincing for the general public? Okay. Obviously, as synthetic biology touches things like origin of life and life systems and so on, we have to be aware that there are some big sectors of the society. I really don't know how this is in India, but in Europe, certainly in continental Europe, people are very, very, very reluctant to talk about genetic engineering and genetic changes and so on. So this is something that, again, I would like to have some feedback from you, not right now, but in the last day where we have this general conversation. Okay. Okay, great. So I think we had a very interesting day today. And now what I'm going to do is to bring my computer, sitting here, do my things, but you are welcome to stay around or to go. And in any case, please start working in these IM projects, those of you who have expressed interest to do it, okay? And let's hope that the last few days will be interesting. The last 2 days, I have to see, because I expect to have more time to tell you more things, but I think you should have at least one class on genetic circuits and all the tools that are available to put together genetic circuits, not only transcriptional factors and these things, but also site-specific recombinases. I mean, there are many, many ways, well, not infinite number of ways, but there are various ways of putting together digital circuits, and you will see how different people have proposed different approaches to have digitalization in logic circuits. And then the other lecture that I want to give you is on metabolic engineering. So metabolic engineering is one of the main, say, topics or what you may call the low-hanging fruits of synthetic biology, because metabolic engineering has already produced things that can be put on top of the table, right? Like plastics, high added value compounds, and other, say, things that obviously reveal and expose the value of synthetic biology. Then there are other topics that I don't know, I would like to tell you, but I don't know if I'll have the time, such as containment circuits, these stories that I mentioned on passing, how to make sure that a synthetic bacterium, a synthetic device, does not escape where it is. I would like to tell you something about ribo switches and use the ribo of RNA-based compounds, RNA-based sequences for making a synthetic regulation. That's another big issue. I would like to tell you a bit about life materials, like life leather, life textiles, life different things. And that's also an interesting thing. So maybe at least at the end, you will have a few snapshots of topics that we may have not studied in detail. But my intention in this course was basically to share with you what you may call the fundamentals. And I'm happy that at the end of the course, I hear your presentations. You talk about devices, about parts, about standardization, so this is exactly the type of fundamental knowledge and fundamental nomenclature and terminology that you have to handle for understanding synthetic biology, regardless of the final applications. Okay, so in that respect, I think that we have made a good job. Okay, great. So that's all for my side for today. I will stay here for a while, and you are welcome to either stay here or to go or to wherever you want. But now we enter into the period of time where you are welcome to work in your projects. Okay. Okay. So see you in a moment.